Dr. Ophelia Venturelli. And then while I'm doing that, Dr. Venturelli, once you're able to, go ahead and share your slides. Okay, so yeah, Dr. Ophelia Venturelli is an associate professor in biochemistry and biomedical engineering, chemical and biological engineering, and bacteriology at the UW-Madison. The Venturelli Lab uses systems and synthetic biology to understand and engineer microbes to address grand challenges facing society in human health, agriculture, and bioprocessing. Dr. Venturelli began her appointment in 2016 after completing a Life Science Research Foundation fellowship at UC Berkeley in the laboratory of Dr. Adam P. Arkin, which is actually where I had the pleasure of working with Dr. Venturelli as a research associate. Her postdoctoral research focused on new synthetic biology strategies to enhance metabolic flux in bacteria and reverse engineering interactions shaping assembly of human gut communities. She received her PhD in biochemistry and biophysics in 2013 from Caltech with Richard Murray, where she studied single cell growth and gene expression dynamics and the role of feedback loops in metabolic gene regulatory networks. Dr. Venturelli has received numerous awards for her multidisciplinary research, including the Shaw Scientist Award in 2017, the Army Research Office Young Investigator Award, also in 2017, as well as a Wisconsin Alumni Research Foundation Innovation Award, the ACS Synthetic Biology Young Investigator Award, and the OVCRGE Early Career Innovator Award just this last year. So yeah, with that, I'll let you take it away.
Thank you so much, Alex. Can everyone hear me? I just want to make sure that
I can hear
there's
you.
There's a lot of background noise. So, okay.
Hopefully
it doesn't come through. Okay, that's great. That's fantastic. Okay, let me just okay, and you can see my slides too?
Yes, they look great.
Okay, perfect. All right, so yeah, thank you so much, Alex and Sean, for the opportunity to give a talk on some of the work that we're doing, studying the gut microbiome and the interactions that shape the system. We're interested in metabolism and also in pathogens. I'm excited to tell you about some of the work today. So I'm sure our gut microbiome has been introduced extensively, but there are trillions of bacteria that have co-evolved with us for a very long time, thousands of years. And this community in a healthy state can break down dietary fibers and produce a myriad of different molecules and degrade molecules that affect our cells, our bodies, and also have feedback loops that affect the gut microbiome composition and functions and metabolic activities. And this is a very complex system with numerous interactions with dietary fibers and other molecules like drugs and things that we consume, and between these bidirectional interactions between the host and the microbiota. So that is in a healthy state. This community is in homeostasis. And then if there is a disruption, say as a pathogen is able to secure a niche, then the community composition and function changes substantially. And that can lead to substantial alterations in the metabolites that are being produced and degraded, which in turn has a profound impact on our health. And this can cause inflammation, and there can be also feedback loops that reinforce this inflammation because these pathogens can actually create ecological niches that they can exploit. And that in turn creates more inflammation and disrupts the gut microbiome. And so this is a very dynamic balance that can be easily disrupted with antibiotics, with drugs that we take, with host dysfunction, and with pathogens. And so we want to understand this dynamic system and ultimately what is driving this system or the interactions between the gut bacteria and also between the host and with the environment. So, you know, this meeting is focused on understanding interactions that affect C. difficile and its, you know, ecological niche. And C. difficile has been introduced already, so I don't need to explain a lot about it, but it's a very deleterious pathogen. And antibiotics are the first line treatment for C. difficile and can be effective. But in some cases, a significant fraction of cases, patients can have recurrent C. difficile infection where after taking the antibiotic, C. difficile comes back and causes, again, this really severe infection. And this can lead to death and severe digestive colitis and inflammation. And one of the remarkable things is that fecal microbiota transplantation, which has been known for a long time, where we can take bacteria from healthy donors and transplant them into patients that have a C. difficile infection, this can be more effective than antibiotics in treating recurrent C. difficile infection. So here we see that gut bacteria can be used as a drug to inhibit a pathogen, which is really a fascinating discovery. And so this is, kind of lays a foundation for the fact that, microbiota, healthy microbiota can be used as a drug, but there are a lot of limitations that prevent FMT from being kind of our future goal. One of them is this transfer of antibiotic resistant pathogens or other unknown factors, because the samples are often not characterized or standardized. And there's a lack of scalability, so there can be a lot of donor variability. And we can't easily personalize this for other types of pathogens or other types of diseases. Using FMT, we don't really know what the mechanism is, and therefore we can't optimize it or personalize it for different individuals with different types of diseases beyond C. difficile. So we want to think about the next type of treatment that we would like to design and understand how to use, replacing FMT with bacterial therapeutics that are defined, standardized, optimized could be a future goal. And these bacterial therapeutics could be as effective as FMT, if not more effective, and consist of known combinations of bacteria that are well characterized. And this could be potentially personalized if needed to different individuals. And also we can understand the mechanisms that by which these bacterial therapeutics work. So this is a really exciting goal, and something that we're working towards, and many others are working towards as well, and pharmaceutical companies are also working towards. But there is a challenge with navigating this enormous design landscape. So when we think about all the different bacteria that we could use to build a bacterial therapeutic, the number is enormous. So within our gut, we have about 100 species, and that would lead to 10 to the 30 possible combinations of commensal strains that we could potentially combine. If we think about including this with other types of fibers or other molecules, then this design space gets even larger. And the way we think about this problem is a landscape where on the axes we have community composition, which is the genetic features of a microbiome weighted by the abundance of different strains, and how that maps to C. difficile abundance or toxin production. We think about this as a microbiome structure function landscape. And if we want to design a bacterial therapeutic, then we want to move the system from kind of suboptimal part of the landscape to a more optimal part of the landscape. So we want to be able to inhibit C. difficile with these bacterial therapeutics. But this landscape can be very rugged, could have had many peaks and valleys, and hard to understand and hard to predict. And we can't exhaustively explore it, even with the most advanced high throughput methods. And so our lab approaches this problem by combining high throughput experiments. We use liquid handling robots that are housed inside of anaerobic chambers. And we also use droplet microfluidics, which is a technique where you can encapsulate single cells or communities into these picoliter to nanoliter emulsions and study about 1 million different single cells or communities in a given experiment. And we use this data to combine with computational models that range in the types of resolution or the types of interpretability of the model to predict and design the system. And we're interested in pathogens, but we're also interested in lots of other microbiome related metabolic functions as well. And so this allows us to kind of efficiently explore this structure function landscape. And we can use techniques from Bayesian optimization, which is a principled way to systematically explore this landscape by balancing kind of uncertainty in our model and our objective, which is, inhibiting C. difficile and in a like wide range of environmental contexts. So I wanted to quickly introduce computational modeling and highlight that there are different tools available. Microbiomes are a unique system. They are very dynamic. So capturing dynamic behavior is important. And also they are kind of driven by different types of interactions, could be pairwise, could be higher order. So we want models that can capture the complexity of these systems. We also want models that give us biological insight out of the system. So if we think about kind of the features of a model, we have flexibility, where the model could adapt to experimental data, or interpretability, where we could extract biological information out of the model. And so genome scale metabolic models are very interpretable. They have high kind of resolution and granularity, whereas the recurrent neural network or machine learning models are very flexible, but they lack that kind of mechanistic kind of biochemical basis. And so we can't easily look at the parameters of a machine learning model and extract biological information, but they're highly flexible. And so two models we've used very widely are the dynamic ecological model called the generalized lock of Volterra. And this model I will kind of mention frequently in the talk. And so I just wanted to point out that this model represents the interactions between different species in a pairwise manner. So it's basically how species A or species I affects species J, and captures the temporal behavior of the community as a function of each species' growth rate and its pairwise interactions with all constituent community members. And so this model can give us insight into how species interact. It lacks some flexibility because it doesn't capture higher order interactions, but it is very interpretable. So we can get information about the ecological interaction network in the system. So there are other models that we are working on to kind of balance some of these trade-offs and also explore and exploit new sources of information. So for example, our microbiome recurrent neural network model is a physics-informed neural network where we have eliminated the possibility of negative species concentrations or spontaneous appearance of different factors in our model, whether that's species or metabolites. And this was recently published in PLOS Comp Bio. And then we also are working on models where we can combine physics and machine learning and also capture the bipartite network that is driving microbiomes between metabolites and species. And so that's an ongoing kind of project in the lab. We also have interest in predicting health relevant functions of the microbiome, such as C. diff inhibitory potential from genomic information. So we're working on that as well. And those models allow us to explore new properties of the system and also allow us to capture the flexibility that is needed because there are so many mechanisms within microbiomes. We don't know what is actually driving them. And so there's a lot of hidden dynamics. So with that, I'm going to kind of dive into some vignettes about C. difficile that we've studied in the lab and kind of explain the role of interactions and how important those interactions can be. So as I mentioned, antibiotics are a first-line treatment for C. difficile. Two commonly used antibiotics are vancomycin, which is very widely used, and it's a cell wall inhibitor and targets primarily gram-positive bacteria. And there's also a DNA-damaging antibiotic called metronidazole, which is actually inactive but converted inside the cell to an active drug. So it's a pro-drug that's activated by anaerobic bacteria into an active drug. And actually metronidazole was used and is used for other types of treatments, infections beyond C. difficile. But now I think it's due to some interesting kind of decreases in efficacy. It's no longer kind of the recommended antibiotic. But we were kind of curious about metronidazole and why, trying to understand maybe why it could be limiting. And so these are two very widely used antibiotics. So we were interested in understanding, could interspecies interactions, we know the C. difficile is part of a complex ecosystem, it's not alone, and so it interacts with the gut bacteria. Could the interactions affect C. difficile's response to antibiotics? And so we know from our previous studies, and I'll talk a little bit about this towards the end of the talk, that gut microbes can be very inhibitory towards C. difficile. And this is evidenced by the efficacy of FMT, that gut bacteria can inhibit C. difficile. So there are these gut bacteria that inhibit C. diff, and these antibiotics can affect the gut bacteria. They also can affect C. diff. So how does that lead to the kind of community assembly dynamics that we see in response to antibiotics, in response to these clinically relevant antibiotics? So we did an experiment in vitro where we took commensal bacteria and we co-cultured these bacteria with C. diff, and we exposed the communities to metronidazole or vancomycin, a wide range of concentrations. And these are kind of like pie charts summarizing our results. That for metronidazole, we found that in most cases there was, slight maybe changes in the MIC of C. difficile, but they were very moderate. And only in a limited set of cases did we see a substantial change, which is kind of the red slice of this pie, in the MIC of C. diff in response to these gut bacteria. For vancomycin, we saw more kind of variation, but we didn't really see, vancomycin, we didn't actually see substantial changes in MIC, but we did see that in the sub-MIC response, which might be relevant when antibiotics are kind of degrading in the gut, or they might not reach the same concentrations in vivo, depending on the tissue and where the local concentration of the antibiotic could actually be in the sub-MIC range. And so we examined the sub-MIC range as well, and found that there was a substantial fraction of communities where C. difficile's growth was altered in the presence of certain gut bacteria when these antibiotics were present. And so this showed that while MIC was mostly pretty resilient to microbial interactions, the sub-MIC response was highly sensitive to microbial interactions. And to kind of highlight one of the most substantial effects we saw, so we know that gut microbes are inhibiting C. diff, and metronidazole is inhibiting C. diff as well. And if we look at the dose response, we see that C. diff is killed at a certain concentration of antibiotic. But when we co-culture C. diff with a bacteria called D. piger, C. diff becomes essentially resistant to the antibiotic. So all of a sudden, C. diff is not killed by any concentration that we tested in this experiment. And so this was really interesting and very unique to D. piger. There was no other bacteria that had this substantial effect on C. diff's antibiotic susceptibility. So we were curious what the mechanism could be. And there's one possibility that D. piger is just degrading the antibiotic directly, which we ruled out with some experiments. There is also the possibility that that there is an indirect interaction where the presence of D. piger somehow renders C. difficile resistant, but it's not directly interacting with metronidazole, it's doing something to C. diff. And so there's one property of D. piger that we know is very notable, which is that D. piger produces hydrogen sulfide, and as a consequence can sequester different metals in the environment. And so we were, given the fact that this was D. piger was the only strain that had this substantial effect, we investigated this using transcriptional profiling, where we co-culture either C. diff alone, co-culture C. diff with D. piger, or we culture C. diff alone. And what we found was that D. piger induces a really substantial metal starvation signature, where certain iron sequestration acquisition genes are upregulated, and then there's iron sulfur cluster binding proteins that are substantially downregulated. And while the mechanism by which metronidazole is converted to an active drug is not known precisely, there are hypotheses that there are certain enzymes that are inside the cell that are performing this inactive drug to pro-drug to active drug transformation. And these genes were actually downregulated. So this suggested a really interesting mechanism of resistance in only a community context, where in C. diff and monoculture, it is producing enzymes that can convert metronidazole from this pro-drug to active drug, and there are metals present, which are required for these enzymes to function. But in the presence of D-Piger sequesters these metals, and that in turn leads to a downregulation, or basically lower activity of these proteins and downregulation at the transcriptional level, which in turn then makes C. difficile resistant to metronidazole. So this was really fascinating to me, that microbial interactions, just a simple co-culture, can enable antimicrobial resistance to an important antibiotic. And this might not just be limited to C. diff, this could be actually more broad to other types of bacteria that are targeted with metronidazole. So metronidazole is used pretty widely for other types of infections. So I just wanted to make sure I stay on time, but I think I have a little bit of time left. But feel free to let me know if I am going over. So there was another really interesting phenomenon we found, which is in the sub-MIC range. And as I mentioned, this could also be physiologically relevant, where in the presence of certain gut bacteria, we saw this alteration in the growth of C. diff in the presence of sub-MIC concentrations. And this was very ubiquitous. This was not a rare phenomenon like the D-Piger scenario. And so there's a hypothesis that due to biotic competition, when there's biotic competitors that are sensitive to the antibiotic and they're inhibited by this antibiotic, that could lead to an attenuation of the competition, which in turn kind of has a competitive release on C. diff. So C. diff could essentially bloom in response to sub-MIC concentrations. And if this is true, then we would expect that the biotic competition between different gut bacteria and C. diff would display an informative relationship with the growth enhancements or kind of bloom of C. diff in response to the sub-MIC concentration. And we actually did see this both in pairwise communities and in multi-species communities, where in communities that have these sensitive biotic inhibitors, they are inhibited by antibiotics, whether it's vancomycin or metronidazole, and that can lead to this kind of bloom of C. difficile at the sub-MIC level, sub-MIC range. So this is also very counterintuitive. It says that in the sub-MIC range, you can actually have, depending on the ecological context, you could have kind of a bloom of C. diff that is unexpected, and this is just due to this competitive release phenomenon. We built a computational model, and this model was very simple. It consists of a generalized local Volterra model that I described earlier, that has an antibiotic susceptibility term, which is BI times A, and A is the antibiotic concentration. And we wanted to ask whether this model, simple model that captures interspecies interactions and monospecies antibiotic susceptibility, could recapitulate the trends in our experimental data. And I should mention that this model is only trained on monoculture data. So it's not trained on any, well, actually it's trained on pairwise data, community data as well, but not in the presence of an antibiotic. So we're really testing to see whether a model trained with information about pairwise interactions and monoculture antibiotic susceptibility could predict the bloom of C. diff at the sub-MIC range. And we found that the model was actually pretty good. And we had a series of null models that we tested where we shuffled parameters to see how, whether it's like, kind of a random phenomenon that our model's working fairly well and has a 71% accuracy in predicting kind of the outcome of the dynamics in response to antibiotics. This model kind of outperformed other null models, showing that this really simple ecological rule could explain whether C. difficile's growth would be kind of enhanced in sub-MIC concentrations in certain communities. So these are kind of two phenomenon that occur, either C. difficile's MIC is substantially altered, or C. difficile has these alterations in growth dynamics that can lead to blooms, and that in turn could potentially have consequences on the host. So with the remaining bit of time that I have, I'm going to maybe quickly, and I just wanted to check, maybe I'll double check how much time I have left so I don't go over too much.
Yeah, you have 5 minutes. You have like 5 to 10 minutes.
5 minutes. Okay, sounds good.
Yeah, even 10 minutes is fine. Yeah.
Oh, okay, sounds good. So now we're going to like a different kind of context where antibiotics are not present anymore in the system, and we want to understand how different bacterial species interact with C. difficile. And our previous work, we showed that C. difficile, it can be inhibited by every single commensal gut bacteria, and the mechanism by which C. difficile is inhibited is through resource competition. We hypothesize certain antimicrobials that we don't know. These are unknown antimicrobials, and also acidification of the environment, which could be potentially relevant in vivo as well, because pH changes a lot during C. difficile infection and inflammation. And so the way that we studied this was using this generalized lockable terra model, where we can combine different commensal bacteria with C. difficile, and then we can use that time series data to fit to a generalized lockable terra model and extract this AIJ parameter, and that's what this heat map shows. So every square in this heat map represents the effect of species I on species J, and we can see that C. difficile is inhibited by every single commensal bacteria. So the C. difficile row is all red. And this is also consistent with a negative relationship between the initial richness of the community, so how many species we combine into the community, and C. difficile's abundance after 48 hours. This is kind of a negative relationship where at a certain richness, we see that C. difficile essentially cannot grow anymore. There are certain low richness communities that also have low C. difficile. So it's not only that high richness communities can be effective, but low richness communities can also be effective. And so there's one kind of major challenge that we have is that C. difficile has an incredibly diverse genome. It has extreme genetic variability. And so we have a collection of strains that were isolated from healthy patients and also patients that were diagnosed with C. difficile infection. And we did whole genome sequencing on these strains and found that their accessory genome is pretty substantial, about 2,743 genes. And there's also a core genome, which all C. difficile strains harbor. And so C. difficile has this enormous strain variability. And so we were really interested in understanding how this strain variability affects interspecies interactions, and also could we identify cases where C. difficile's interactions could be C. difficile, diverse strains of C. difficile are inhibited by certain gut communities. So we could actually design robust communities that inhibit many strains of C. diff. And so we designed actually 2 media environments. One of them is a healthy gut media where there's a lot of competition, and a media that we call the perturbed gut media, which has nutrients that C. difficile can exploit in the inflamed gut. And so we compared C. difficile's abundance, and we looked at a wide range of strains. Here I'm only showing four different strains of C. difficile. And on the y-axis, we have C. difficile's abundance, and the distribution is over the different consortia that we built. So we have a wide range of different communities that we built from the bottom up using our isolate collection. And so we can see that overall C. difficile's abundance is substantially higher in this perturbed gut media than in the healthy gut media. And so we were interested to know, how this kind of changes interspecies interactions. So when we took the data and fit the data to a generalized lockable terra model, we found that in the healthy gut media, there were, again, lots of negative interactions. And I'm sorry, I'm de-identifying some of these species because we're currently kind of submitting invention disclosures and working with our WARF on campus on this. So, but yeah, so anyway, we found basically lots of negative interactions in this healthy gut media, as consistent with our previous study, that there can be many negative interactions that inhibit C. diff, but in this inflamed gut or perturbed gut media, we found only one strain that had a really substantial negative interactions with C. difficile. And it was the only strain that could inhibit all C. difficile isolates. And I should also mention that it also inhibits toxin production for all strains. So it is a very unique species and a really unique interaction. And we also decided to use our computational model to guide the design of certain communities. We picked two in this case. One of them has some commensal bacteria that are predicted to have a weak inhibition, which is WIC. And then we also have a community that has this unique species and another species that we identified. And these two, we have the strong inhibitory community and the weak inhibitory community. And so we used the model to actually select these communities, and we wanted to test to see whether in mice, could these communities protect C. difficile from C. difficile challenge. And sorry, actually, I think my animation kind of broke here. And what we found actually was that when we take these two, the weak inhibitory community and strong inhibitory community, and test it in vitro, the weak inhibitory community allows C. difficile to grow to a substantial level, very high level, whereas a strong inhibitory community reduces C. difficile's growth. And so and this is across multiple strains. And so we took these communities and tested them in mice. So we gavage mice with C. difficile that had these two different communities. And we found that the strong inhibitory community can actually protect mice from C. difficile challenge, whereas the weak inhibitory community, all the mice died from this pathogen. And so this was really fascinating. So we wanted to understand what was unique about sorry, and I think some of my animations are kind of broken here, so I'm having to fix this. Yeah, so one thing that's actually pretty unique about these two species, in particular CL2, is that it has a very high metabolic niche overlap with C. difficile. This was unique to all the other species in our community. And it essentially eats almost the same nutrients as C. difficile. So it has the highest carbohydrate utilization overlap. It also has strict amino acid metabolism. And so we were interested to know what was unique about this particular species. And I'm sorry, I'm just gonna have to zoom out here because for some reason it's blocking it. So we did an RNA-seq experiment where we have our species and we co-culture it with C. difficile, or we have C. difficile alone, and we looked at the gene expression changes in co-culture. And we found a massive change in the metabolism of C. difficile in response to this species. And this was very unique. If we look at other closely related species to CL2, they don't have this same effect. So there's something very unique about this species that is causing a massive change in metabolism. And this extent of metabolic shift was not a very specific shift in specific metabolic pathways, but it was throughout metabolism. And so this, we don't really know exactly the answer about the mechanism. There's several different hypotheses, but it does seem that the substantial niche overlap that this bacteria has with C. diff is involved. And there could potentially be other mechanisms that play as well. So in summary, I've shown that negative interactions are very sparse in environments that contained C. difficile preferred substrates in the inflamed gut. And we found a species that has the highest degree of niche overlap, and its inhibitory effect is enhanced in communities. It by itself, I think may have limitations in robustness, but when it's combined with other species, it actually can be very robust. And in vitro, it can inhibit diverse C. difficile strains and also toxin production and ameliorate C. difficile infection in mice. And we hypothesize that there is a massive metabolic shift that is taking place that's very unique to this particular interaction. And so with that, I will mention that we're doing, we're very interested in kind of scaling up our measurements, and we've been working with droplet microfluidics as a method to capture single cell phenotype, genotype, and also community as well, and looking at interactions. And we've published a paper earlier this year. We have one more in revision. The idea is that we could eventually take even higher throughput measurements to study communities and their interactions with pathogens or other types of functions. And this could allow us the ability to look at millions, tens of millions of communities simultaneously. So really excited in the future, implementing these methods to identify these unique communities that are, anti-pathogen or have interesting metabolic functions. So in sum, I would like to thank our lab, which is a really diverse group of scientists and postdocs, our collaborators at UW-Madison and also outside of UW-Madison, and our funding sources. And I'm happy to take any questions. Thanks.